All right, last page of 10.1. So we finished monohybrid crosses on the last page. So these are going to be called dihybrid because they study two traits at once. So we're going to look at if I crossed, like a pea plant that was tall and produced purple flowers with one that was short and produced white flowers, what are all the probabilities of those combinations? So these are going to be more complicated. They're going to be 4 by 4 and have a total of 16 boxes. So each box is actually only worth like 6.25%. So this is when it's sometimes a lot easier to actually go with the fraction on these. So begin again. Above and to the side, instead of having one allele, one letter, we're going to have two alleles, one for each gene that we are discussing. So we'd have one letter for the height gene, one letter for the color gene. So to figure out all the possible alleles, we're going to steal from math class and FOIL. Remember FOIL, first, outside, inside, last. Okay, so here's an example. So if a plant is heterozygous for height and seed color. Remember, heterozygous means they have two different alleles. So it'd be big T, little t, big Y, little y. So when we FOIL, we go the first of each letter. So we're going to have big T, big Y. The outside letters, big T, little y. The inside letters, and the last of each letter. Okay, so first of each letter, outside, inside, last of each letter. And these are the 4 possible combinations that we could put into gametes. Okay, so if this was my parent, 25% of them would have the tall allele and the yellow allele. 25% would have the tall allele and the green allele. 25% the short allele and the yellow allele. And 25% short and green. So you do this for both parents, and these are what go down the side and across the top. Okay, so this is one parent. Our other parent is big T, big Y, little y. So I go first of each letter, big T, big Y, outside, big T, little y, inside, and last of each letter, like that. Okay, so see if my whole Punnett square fits. So it does. Alright, so down the side, I'm going to do my parent 1 down here. So my parent 1 possible combinations were big T, big Y, big T, little y, little t, big Y, little t, little y. Second parent, remember, so this was first, outside, inside, last, big T, big Y, big T, little y, big T, big Y, big T, little y. So when we fill these out, priority 1, keep the genes together. So I'm going to keep the T's with the T's and the Y's with the Y's. That's top priority. Second priority is if you have a big and a little of the same gene, like a big T and a little t, we want to put the capital T first. But anyway, so over here and up here, so we're going to put all 4 of these letters in this box. So I'm going to put the T's together and the Y's together. Going down. So I want up here and over here. All these letters in here together. Big T, big T. And now I have a big Y and a little y, so I want to put the capital first. I would really encourage you to hit pause here and try to fill out the rest of your Punnett square, but I'm going to kind of just go as fast as I can here. And double check with mine when you're done. Notice that none of my letters would you look at and be like, is that capital or lowercase? I can't tell. Right? They're all very clear what's what. Okay. Oops. Alright, so then, if I was going to figure out possible phenotypes and genotypes, okay, there's a phenotype, big T, big Y, big Y. So genotypes, big T, big Y, big Y. So I go and I say, well, how many of those do I have? Okay, so I have, there's one. Is that it? I do not see any more. So I would say there's a 2/16 chance. I know I could reduce that, but I'm going to choose not to because then I'll have a common denominator in 16 and it's going to be easier for me to compare. So there's another genotype. Let's do this one. Big T, big Y, little y. So again, I'm going to say, how many of those are there? So there's one, there's one, there's one, and there's one. So this one has a 4/16 chance. Let's see. Here's another one. Big T, little y, little y. I see there's one there. And there's one there. And that's it. So there's a 2/16 on that one. Whoops. And then we have the big T, little t's. So big T, big Y, big Y. There's one. There's one. 2/16. Big T, little y, big Y, little y. There's one, two. This one should be like that. 3, 4. And big T, little y, should just be 2 left. 1, 2, 2/16. So there's the probability of those genotypes. Then we have the phenotypes. They're not going to be nearly as many because remember, you just need one dominant allele for a gene for that version of the trait to show up. So they could be tall and yellow. Okay, so they have to have at least one capital T and at least one capital yellow. They will be tall and yellow. So these 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 out of 16 will be tall and yellow. Or you could look at what you already counted up. So these will be tall and yellow. These would be tall and yellow. These would be tall and yellow. These would be tall and yellow. And these would be tall and yellow. So that's 4, 5, 6, 7, 8, 9, 10, 11, 12. Then they could be tall and green. So this would be, they have at least one capital T, but 0 capital Y's. So that would be 1, 2, 3, 4. And that's it. Right? 12 and 4 adds up to 16. So that's it. You do have a packet of practice with these dihybrid squares. Remember, as always, please let me know if you need any help on those. See you later. Bye.